Very good morning and very good day. So today I'm going to share a little bit about what our lab has been working on for the past few years. So it's about cytotoxicity of antimicrobial peptides from the skin mucus of anabas testudius on breast cancer cell lines. So these are the teams who are involved in the whole project. So we are talking about AMPs or antimicrobial peptides. So one of the best sources of AMPs are from mucus from the fish. So from our studies, we actually carry out and pick up one of our local fish called or what we call ikan puyuh. So we would like to test the whole antimicrobial peptides of the mucus to see whether it works as an antimicrobial peptide, as well as to see whether it does have an anti-cancer effect on the cancer and breast cancer cell line. So the hero of my studies today is basically this fish. So this is what we call ikan puyuh or puyuh fish, the climbing perch, which are locally available in Malaysia. So the reason why we pick up this fish, as we can see in the slides here, these fish are pretty much robust, and they have a very thick mucus that encode the whole fish to actually protect them protect them from a lot of stuff. So you can see here they can actually walk on land. So these are the robust how robust the fish really is. So what we did in our study is that we actually challenged the fish with microbes. Basically, the inert microbes that has been autoclaved or killed, and then we actually treat the fish in the tank and we just scrap the whole mucus of the fish and did a series of mucus extraction. We did a protein fractionation, identify the antimicrobial peptides, and try to identify the pathway that affect the anti-cancer cells. So I won't tell all of this result in here, but we'll try to brief through the whole project, right? So from our challenge, what we actually found is that this particular fish, once we actually treat and challenge them, it increased the mucus production. So as you can see here, the control, the whole total protein with the control fish is much lesser compared to the challenge fish. So this is the first time we actually developed this method where we can actually increase the mucus production of the fish when we actually challenge it with dead bacteria. So we challenged it for 14 days, and you can see that it increased throughout the challenge treatment. So this is the crude mucus antibiotic activity. So we actually tested back with the same bacteria that we had actually used to challenge the fish. And you can see here that the the AMP or the antimicrobial peptide does inhibit the the bacteria. So you have E. coli, Pseudomonas aeruginosa, Bacillus staphylococcus, and Bacillus cereus. So these are the things that we actually found in the whole project. So after that, what we did is that we took the crude extract, we actually run through the cell line. So MCF7 is a breast cancer, MCF7 and MDA is actually a breast cancer cell lines. MDA is basically a triple knockout of breast cancer, triple negative. Sorry. So MCF7 is basically is a breast cancer that pretty much involved with hormones and stuff. And HS27 is our normal fibroblast. So as you can see here, that the mucus that we extracted from this particular cell, from this particular mucus, does inhibit cancer cell lines. So what we did after that, we actually run through a Cephedex and try to isolate these particular fragments or other proteins within the mucus. So we actually found four fractions. We managed to actually get four fractions from this particular mucus. So we actually pick up this fraction. Fraction 1, fraction 2, fraction 3, and fraction 4. So also known as F1, F2, F3, and F4. So what we did, we do the experiment again with the bacteria, and we actually found the same thing as well. So what we found is that the best fraction to inhibit the bacteria is fraction 2. So this is what we actually found in all the bacteria. So we also repeat the same experiment with the breast cancer cell lines, but we found that in fraction 2, it does inhibit MCF7 and MDA cell lines, while it does not affect HS27, which is a normal fibroblast. So what we did after that, actually run to a flow cytometry stain to see what's the effect of the fraction 2 onto the cell cycle on all three cells, HS27, HMCF7, and MDA cell lines. So you can see a plus T and minus T is actually without the treatment. So you can see that on the left images, you can see that during 6 hours to 48 hours, you can see the how normal cell lines progress in the cell cycle. So with or without treatment, you show the same profile. While without on the MCF7 and MDA cell lines, what you can actually see here is actually there is a perturb of cell cycle or increase of cell cycle checkpoint, especially in G1 on treated cell on both MCF7 and MDA. So in compared to the without the negative treatment, which is much normal flow of cell cycle. So what we did after that, we actually did, we tried to identify what kind of peptides that exist in the fraction 2. So we did LCMS identification. So we found 55 peptide. And from the 55 peptide, we actually run through a certain analytical model, ADAM, CAM3, P2, I, IM2L, ADP3, AMP5. So we try to cross-link with all the peptides that might interact with any other cancer pathway. So we actually found two peptides we actually responsible for this, which might have some sort of effect on anti-cancer. So it's peptide 1 and peptide 2. So we run back, we get this peptide, we know the sequence, then we actually synthesize this peptide, the first peptide and the second peptide. Then we run through the same experiment again to see whether it does inhibit the bacteria. So it proved it does inhibit, especially on the second peptide. Then we run through again with the to do a anti-cancer cytotoxic assay, MTT assay. So what we found, it does both peptide actually inhibit the cell lines or the breast cancer cell lines compared to the HS27, which is a normal cell line. So it does correlate with the previous findings. So these are all the sequence that we found. So these are some of the analysis that we showed here, so which I will not elaborate here. So we run the same flow cytometry test. It showed the same thing as well, where the first peptide and the second peptide does inhibit the MCF and MDA cell lines at the G1 pathway. So you can actually see the second, especially on the second peptide, where you can actually see that it does some sort of increase the cell signaling checkpoint on G1 pathway. So what then we actually try and we run the whole peptide again, and we do some sort of DNA fragmentation assay to see whether it does increase apoptosis in these cells. So as you can see, the HF77 cell line does not have any DNA fragmentation as compared to the MCF7 and MDA cell line were actually treated with this peptide. So you can see the increase of DNA fragmentation of all these cell lines. So this indicates that there are apoptosis. To prove this further, we run an annexing FIT-C apoptosis assay. So we have the control here, which is HF27. So after a prolonged with the control first peptide and second peptide, they say that there's a lot of viable peptide, not much of a late apoptosis or early apoptosis cells. But when you compare to a first peptide and second peptide treatment with MCF7 and MDA, we can actually see that there are increase of late apoptosis of cell lines in these two cells. So we run a gene expression PCA array in these cells. So what we found is that there was an increase of a lot of gene marker for, especially for apoptosis, such as BEX, caspase-3, caspase-7, P53 cell line, protein throughout all of these two cell lines, MCF7 and MDA. And interestingly, BCL2, which is an anti-apoptotic protein, is also downregulated, which proves that this particular peptide does induce apoptosis. So we done a sort of a mini in silico studies. We've tried to actually do a protein-protein interaction with this particular first peptide and second peptide, and it does show that it's bind to this all this apoptotic protein to this peptide. So the first caspase, it binds to caspase-3, caspase-9, P53, and BCL2. So as we all know that all these particular peptide, it does involve in apoptosis pathway where caspase-3 and caspase-9 and P53 is more control in upregulated in terms of to actually induce apoptosis, while BCL2 does reduce reduction of BSL2 increase apoptotic activity. So this is basically what we try. We hypothesis is our hypothesis by saying so. Part of the study has not been we still is still being carried out in the lab. So we do see that the first pattern induces cell death of MCF7 and MDA cells via intrinsic apoptosis pathway by modulating the expression of apoptosis-related genes P53, BEX, BCL2, caspase-9, caspase-3, and caspase-7. P53 also regulated in transcription of the cycle-dependent kinase inhibitor P51, which promote G1 to S checkpoint arrest. So we do believe that these are the things that happen to the cells when we actually try to actually induce apoptosis in these particular cells. So we are happy to announce that we actually just published these findings. Feel free to actually look through this paper. So, yep, thank you very much.